So next we have is our rods. Okay, so before understanding our rods, let's just quickly go through all the things, okay, that will lead us to the understanding of our rods. So first we have the multipotent hematopoietic stem cells. Where is this? This is present in the bone marrow. From this one particular cell, all the cells are generated. Okay, so all the circulating cells in our body, maybe it is RBC or platelets, mast cells, all the WBCs, granulocytes or agranulocytes, macrophages, everything is coming from this multipotent hematopoietic stem cells from the bone marrow. So our target will be not all of this, only from here to here myeloblast, and from there to here neutrophils. Why? Because see, the our rods, that is primarily found in something called as acute myeloid leukemia. So you leave the acute, leave the leukemia, just myeloid if you take. So that means he's essentially talking about the myeloblast, and among the myeloblast, the neutrophils are huge in number, around 50 to 70% is neutrophils. Okay, so this is our primary focus. So multipotent hematopoietic stem cells, we will be talking about myeloid cells and then the neutrophils. Then you need to know about leukemia, because as I said, acute myeloid leukemia. Myeloid, you understood. Okay, what is myeloid? That cells. These myeloid cells, the myeloblast, all these myeloid cells. Okay, myeloid progenitor cells. So essentially that is what we will be talking about. Now what about leukemia? What is leukemia? Leukemia is nothing but blood cancer. That means all these cells, or maybe these cells, either it is this or these cells, will be huge in number. Generally WBCs are around 4,000 to 11,000 in number in healthy individual. Okay, but in case of leukemia, it crosses like 50,000 or 1 lakh. Okay, in cell number. So there is huge proliferation of these WBCs due to some abnormalities or genetic mutations, which is not normal, not healthy, that is pathological. So we understood leukemia means blood cancer. In this blood cancer, the WBCs are proliferating excessively in number. Then the leukemia has two broad classifications. One is chronic, the other one is acute. In chronic, that means for the prolonged period of time the disease is ongoing, and acute means like recently it has started. So recently maybe the cells have started in proliferating huge in number. Okay, so this is more severe form. Acute is the more severe form. So we will be discussing about this part, not this part. So leukemia, acute leukemia, then we will be talking about acute myeloid leukemia, not the lymphocytic, not chronic, because the all rods is found here in the acute myeloid leukemia. Okay, then look at this. So myeloblast, where did you see this? Here, myeloblast. Where it is coming from? Hematopoietic stem cells. So it is going here and not here, it is coming here. Myeloid progenitor, myeloblast, neutrophils. Why are we talking about neutrophils and myeloblast? Because we are talking about acute myeloid leukemia, in where these cells are proliferating huge in number. Among that, most is neutrophils, because that is abundant in number, already present in the body. So myeloblast is getting converted into promyelocyte, then myelocyte, metamyelocyte, band cells, mature neutrophils. So this is the normal developmental stages of the neutrophil. So what goes wrong in the acute myeloid leukemia? In acute, because it is acute, then it is myeloid means these kind of cells, and leukemia means huge proliferation of the cells. So in acute myeloid leukemia, so myeloblast and promyelocyte, these two stages will be there in the blood in huge number. Okay, so there won't be any maturation or differentiation, won't be there due to some genetic abnormalities or translocation that we will see further anyways. So in acute myeloid leukemia, if you see the cells under the microscope, there will be big cells compared to the mature neutrophils. See, in mature there are 3 lobes. Okay, but here can you see any lobes? No, there is only one nucleus, big nucleus. Okay, and note down the color is like blue, shades of blue or purple kind, right? So myeloblast, promyelocyte, here is where you will find the AUER rods, not here. Here generally you don't see R rods, but here you will see. Why? Because in acute myeloid leukemia these cells are not formed, these are only there. These cells are not able to differentiate into all this, they are not able to mature. So as you will see these cells more, there are certain enzymes in these cells, okay, called as peroxidase enzyme, okay, myeloperoxidase. Then there are other enzymes and other proteins formed at this stage. So all this will clump together. Why they will clump together? Because naturally as they mature, those proteins they diffuse everywhere in the cytoplasm, okay, as they mature. But because there is no maturation, those proteins or myeloperoxidase enzymes or whatever it is there at this stage, they tend to come together and clump together, and then they form something called as rods. Okay, they are like needle like azurophilic granules. They are needle like or rod like. What is the meaning of azurophilic? Azuro means blue color or shades of blue or maybe purple color. Philic means loving. So azurophilic granules means at this stage there are certain enzymes or certain proteins or certain granules are there at this stage, okay, which are blue loving. So distinct needle like azurophilic granules are present in many cases. They are particularly numerous in acute myeloid leukemia with the translocation of 15th and 17th chromosome, which is nothing but the subclassification is acute promyelocytic leukemia. Now I understand you understood till here. Okay, distinctive needle like azurophilic granules are present in many cases. Okay, then you everything might have gone above the head. Okay, they are particularly numerous in AML with what is T15 and 17, what is acute promyelocytic leukemia. So we'll look that also here. So in acute myeloid leukemia, you understood about acute myeloid leukemia. So everything is coming from multipotent stem cells, from there the myeloid cells, from there the neutrophils. Then we have leukemia means two types, acute and chronic. Acute may we have acute myeloid leukemia, acute lymphocytic. We will not talk about all this because the rods are found in AML, acute myeloid leukemia. From there we understood that myeloblast and promyelocyte, these are the early stages of the neutrophil maturation. So these cells won't be able to mature in this form. That is why the granules or the enzymes or whatever is produced at this stage, they won't be able to diffuse into the cytoplasm. Rather they will clump together to form certain rod called as R rods, and these are nothing but the needle like azurophilic granules present in the AML with the translocation of 1517 and acute promyelocytic leukemia. Okay, now so acute myeloid leukemia, if we classify this, there are two classifications. See, one is this called as FAB, French American British classification for the AML. The other one is called as the WHO classification. So in both the classification, in this classification we have 1, 2, 3, 4, 5, 6, 7, 8 different classification types. So we have M0, M1, 2, 3, 4, 5, 6. Out of this, M0 means undifferentiated, AML without maturation, AML with maturation. Then we in M3 we have acute promyelocytic leukemia. See what he's telling here? Acute promyelocytic leukemia. That means he is talking about FAB classification, under which that is M3. M3 acute promyelocytic leukemia. Now if you go in elaborated part, M3 promyelocytic that have hypergranular with many R rods. Okay, so if you stain with certain stains, you will see R rods. See here, you can see, let me just zoom in. See how nicely these rods are present. Here you see this tiny rod here, blue color, right? So these are R rods. So if you have not understood the acute promyelocytic leukemia, this is nothing but people have made classification. One classification is this FAB, French American British classification, under which there is M3, under which there is acute promyelocytic leukemia. Here you will see most number of the R rods. Then there are other like acute myelomonocytic leukemia, acute monocytic leukemia. These are all different things, megakaryocytic. Okay, but in this acute promyelocytic means this stage. So first is myeloblast, then it will convert to promyelocytic, then myelocytic, then so in case of when it is growing, when it is trying to grow into a mature neutrophil, but it is not able to do so. Okay, it is somewhere here, it is stuck. Okay, so why is it stuck? Because of this gene mutation, the translocation of the 15th and 17th chromosome. Okay, So here you see, now in this classification, in WHO classification, they have nicely explained AML, acute myeloid leukemia with T15 and 17. Let's just zoom in. T15 and 17, okay, RA PML fusion gene, okay, that is some retinoic acid or something. So here then there are numerous OR rods, often in the bundles with individual progranulocytes, primary granules usually very prominent but inconspicuous. In microgranular variant, high incidence of the DIC. Okay, so here as you can see, then here also you can see, but not as much as here, the AML with T15 17. So this is very specific. They have mentioned that at this translocation there will be numerous OR rods. This is the classification of WHO. Same thing, the classification in the FAB, they are telling it as M3, acute promyelocytic leukemia, and here Robin has combined both of them. So OR rod is distinctive needle-like azurophilic granules present in many cases, but they are particularly numerous in acute myeloid leukemia with 15 and 17 chromosome translocation, and under FAB classification, acute promyelocytic leukemia, which is M3. Alright. Yeah, so for the 3 marker, what can you write? OR rods definition: rod shaped azurophilic. Azurophilic means blue color loving granules found in the myeloblast or promyelocyte. So this is myeloblast and promyelocyte. So in these two stages you can find OR rods. Not here. It is generally not seen here. Associated with acute myeloid leukemia. So we have two kinds: chronic, we have acute. Acute may we have myeloid leukemia, then lymphocytic leukemia, okay, lymphoid leukemia. So here we are talking about myeloid series, because all this neutrophils, everything is coming. The why myeloid? Because myeloperoxidase is in this cells, okay, myeloid cells, myeloperoxidase in the neutrophils. Those all clump together to form azurophilic granules, that needle-like structures. Significance indicative of the myeloid lineage in the leukemic cell. Okay, so that is about the what? OR rods.